One of the last ways we can divide creatures or take a look and categorize different animals is in their basic embryonic development. We have something called protostome and deuterostome development. Based on early development, many animals can be categorized as having either what we call protostome or deuterostome development. In both cases, you'll have the blastula begin to infold, like we talked about, called gastrulation, and you'll form an interior tube. Now, initially, you'll start with a single opening called a blastopore. We'll talk about this in a little bit further detail. So, at the beginning, you have basically a ball of cells that has folded in on itself and formed sort of a U shape. So, there's only one opening to the outside called the blastopore. Now, through further development, there will become another opening on the other side eventually. But that first opening, the blastopore, that's either going to become the mouth of the creature or it's going to become the back end, the anus of the creature. And those are two different forms of development. If that first opening, the blastopore, becomes a mouth, it's called a protostome. And if the first opening becomes the anus of the creature, it's called a deuterostome. So let's talk just a little bit about those two different forms of early embryonic development. Here's a basic comparison showing a couple of different things that we need to look at. One of those is in cleavage, in that early cell division phase. There's two different ways that cleavage can occur, and we'll talk about that in just a second. One of them is called spiral and determinate, and the other is called radial and indeterminate. So when the cells are initially dividing in the early embryonic development, when it starts off as just a single fertilized cell and then it begins to go to 2, 4, 8, 16, well, we have different forms of cleavage. We'll talk about those. And then here's that coelom formation. When we form that interior cavity, how does that exactly occur? When that infolding occurs and you have that one opening called the blastopore, does that one first opening become the mouth of the creature? If that's true, well, that's called protostome development, and that would be some of your more, I guess you would call them less advanced creatures like your mollusks and your annelids. But if that first opening, the blastopore, becomes the anus of the creature, that's going to be your echinoderms, your chordates. That would include mammals. So humans go through this type of coelom formation, which is called deuterostome development. So the very first opening of the blastula, called the blastopore, in humans and other mammals, that actually becomes the anus of the creature as opposed to the mouth of the creature. And then finally, the fate of the blastopore. What did that become? Well, in some creatures it becomes the mouth, and then eventually there's another opening that becomes the anus. And in deuterostomes, that first opening is the anus, and then the mouth is the opening that is formed later in development. So let's kind of step through that just a little bit, just to get a few quick details, and that's where we'll finish. So first is in cleavage, which is that early cell division. In protostome development, cleavage is spiral and determinate. When we say determinate, we mean the cells are locked in very early on and they can't really change. When the embryo is first developing, a lot of these cells are stem cells or germ cells. They can become almost anything they need to become. So they have not yet become skin cells, muscle cells, nerve cells. They are germ or what we call stem cells that can go on to develop and become almost anything they need to be. So if we're talking about determinate cleavage, what we mean is that those cells become locked into their role early on, and once that happens they cannot change. So in protostome development, which is again perhaps the less complex animal creatures, the cleavage is what we call spiral and determinate. In deuterostome, which would include the more advanced creatures perhaps like reptiles, birds, mammals, the cleavage is radial and indeterminate. In indeterminate cleavage, we mean that those cells, they retain the capacity to go on and become a variety of different things. So they retain the capacity to develop into a complete embryo. Indeterminate cleavage makes possible, for example, identical twins, because if humans had determinate cleavage, once that ball of cells reached a certain size very early on, it couldn't be subdivided. But with indeterminate cleavage, you have a mass of fertilized a mass of cells that sometimes become detached from each other, and then those two separate masses can then go ahead and complete embryonic development and become two full babies. And that's what allows for, say, identical twins. And that's a good example of proof that humans undergo what we call indeterminate cleavage. So here are the two different types of cleavage. Spiral is describing the way that it shapes. Radial and spiral describe the way that they shape. Determinate means the cells become locked in as to what their role is early on, and indeterminate means that they stay pliable and able to change for a longer period of time. This would be your less complex animals. This would be more than likely your more complex animals. Next is coelom formation. How does that interior cavity form? In protostome development, the splitting of solid masses of mesoderm forms the coelom. In deuterostome development, the mesoderm buds from the wall of the archenteron, which is kind of a pouch which forms the body cavity. And again, it's just two different types of ways of developing. So you see you have kind of two masses of mesoderm here, and in the deuterostome, the mesoderm kind of basically becomes a bud off of the interior endoderm and then forms separately from that. So just two different ways of development as far as how does that interior cavity and the mesoderm develop. Finally, the fate of the blastopore, which is that single opening that first occurs in gastrulation. The blastopore forms during gastrulation and connects the archenteron to the exterior of the gastrula. If you are a protostome, from the Greek word protos, which means first, and stoma, which means mouth, so protostome kind of says first mouth, that opening becomes the mouth. If you are a deuterostome, from the Greek word deuteros, which is second, and again stoma, mouth, the blastopore becomes the anus. So in a human, the anus of the developing embryo forms first and the mouth forms second. In a dog, in a bird, in a reptile, the same thing occurs. In a more simple creature that we're going to take a look at in the next chapter, in the early embryonic development, the mouth forms first. So I know for me, this information was somewhat confusing. So I would highly suggest if you go and take a look at animal development, the crash course under animal development does a really good job of explaining these various layers of cells and some of the basic information that we find in this chapter. Let's take one more quick break and we'll get to concept 32.4, and that'll be how we end up this chapter.